I'm going to present some new data from my lab in the second half of the talk, but we're in an art gallery right now. And this is the first ever picture book using a microscope. Famously, Robert Hooke, 1665, drew these kinds of pictures in a book that Samuel Pepys called was the best book he ever saw, completely ingenious. And clearly, microscopes open up worlds that we didn't know existed. So this is not hypothesis-driven research. This is pushing forward technology to open up things that we had no idea that were there. And so this was the first time that people saw a flea looking as monstrous as that. And of course, it became a huge bestseller and is known many years later. So clearly, using microscopes to zoom in better and better and better and better opens up new things. But in 1874, a mathematician, Ernst Abbe, calculated that you cannot use a light microscope to zoom in forever. And that is now the formula that he came to is now inscribed on a stone in Jena where microscopes were initially developed. And essentially, you cannot get a light microscope to zoom in better than about 200 nanometers, half the wavelength of light. This light in this room is roughly, 500 nanometers. And so you can't see better than around 200 nanometers with a light microscope. All the time I've been in Manchester, the last decade, we've been using microscopes that beat that fundamental law of physics. And what's wonderful to me is that the journey that got us to microscopes that beat a fundamental law of physics goes right back to this guy, Osamu Shimomura, with his family, where every summer he went with his wife and 2 children here. He's since passed away, so I got this photo from his daughter, who's the cute girl in the red jumper there. And they went 90 miles off Seattle to Solomon Islands collecting jellyfish with those nets. Osamu Shimomura was interested in how animals and sea creatures communicate with each other through color. And of course, that is quite fascinating. We have very little understanding of how what animals and sea creatures are saying to each other through the use of color. But he was also interested in, how are sea creatures generating these elaborate colors in the ocean. Here he is now. He roped in some other people friends and people hanging around people would go up to him and say, "Oh, I didn't know you could eat these jellyfish. How do you cook them?" And they had to say, "Oh, no, we're collecting these jellyfish for scientific research." Osamu Shimomura, I think, is quite an inspiring character, in part because of how he did science. He had a childhood of some adversity. His father was away a lot, being in the army. He was about 15 kilometers away when the Nagasaki bomb went off. So I think he learned to be strong and independent. And his approach to science came directly from that, in that he would not necessarily sort of read other people's papers and kind of extend them. He would not go to the stockroom of the university to find the reagents he needed for his experiments. He would wander to the market, go to the hardware store, find materials that he thought might be able to filter out bits of cells better than anything else he'd get in the and he said that. He said that himself, that the most important thing to doing great science is learning how the process of how to do science in a kind of independent way. So, as many of you know, he was looking at these particular jellyfish that were first identified in 1955, and he was looking at those cells that underneath the umbrella glow green. He was then isolating components, fractions of those cells to find out what were the things that really glowed green. And he isolated 2 protein molecules, one that absorbed blue light and another then took in one that gave out blue light when it bound calcium, and another protein that took in the blue light and gave out green light. And that second protein is, of course, what we all know as green fluorescent protein, or GFP. Understanding how jellyfish glow green is not obviously on the path to a Nobel Prize. And I think that's also important, that this is curiosity-driven research, thinking about why the jellyfish glow green, and yet, of course, it's changed every aspect of biology that we know today. So that was in the 60s and early 70s, and the story then jumps forward to New York in 1989, where Martin Chalfie is listening to a talk in Columbia University, and he hears someone happen to mention this, there is this GFP, green fluorescent protein from jellyfish, and in his words, it was a light bulb moment, a visceral experience. He realized that he could use this green fluorescent protein because he thought maybe he could get other kinds of organisms to use that protein and get them to glow green. It was before Wikipedia. What do you do to begin this stuff? You start phoning people up. So he's phoning around, anyone heard about this GFP? How do I get it? He was led to phone Doug Prasher, who was working in Woods Hole in Cape Cod. Some of you will know it's a wonderful, relaxing, beautiful place to do science. He phoned Doug Prasher, and Doug Prasher said, yeah, I'm working on isolating the gene for the green fluorescent protein. So Martin said to Doug, great, when you isolate the gene, tell me, and then I'll love to do some work on that. Doug said, great. They then lost touch because Martin Chalfie went on a sabbatical for a year, and Doug Prasher assumed he'd just left science. And then Martin never heard from Doug, so just assumed he'd kind of never isolated the gene. When Martin came back, we're going to come back to that moment in a later. When Martin then came back from his sabbatical, he read a paper by Doug Prasher where he did isolate the gene. So he then contacted Doug, said, oh, I thought you were going to send me the gene for the jellyfish protein. Doug said, oh yeah, sorry, here it is. Martin passed it on to his student, Gia Uzkirchen, who then took the gene for the jellyfish protein and the green glowing jellyfish protein and put it into bacteria. She then looked down the microscope. Has the jellyfish green glowing protein made bacteria glow green? No. Right, that doesn't work. Wait a minute. The microscope in our lab is actually a bit rubbish. Let me just try another one. So she went down the corridor and, oh, actually, wait, they are glowing green. So that was the first time that the GFP was seen to work in a different organism, and that was not obvious. There could have been anything about the environment of cells in a jellyfish that made that protein glow green, but it did work on its own in bacteria. The next bit of the story is a technician in Martin Chalfie's lab, UAN2, who took the same gene from the jellyfish, and this time he attached it to a promoter that makes the gene only switch on in the motor neurons of worms, injected that gene into the gonads of worms, then the offspring all had the gene with the promoter to be in motor neurons. This is the first picture he saw of green glowing cells within the motor neuron of worms. They first announced this result in the Worm Breeders Gazette in 1993, and of course later famously published it in Science. In the Worm Breeders Gazette, they said anyone who wants the gene, you can get that gene. This is, of course, what got Martin Chalfie the Nobel Prize for this work, showing that you can now make cells glow in an organism by using the green glowing jellyfish protein. Martin Chalfie, when I talked to him about that moment, obviously for all of us now, that is a seminal contribution to what we do. But at the time, Martin says no one appreciated it. He would enthusiastically tell people in Columbia University, and they were like, oh, I don't know, it sounds all right. And he says that basically when you do something that's really different and really new, technologically important, making an advance, no one gets it. It takes quite a long time for people to understand the importance of what you've done when it's really new, and you have to tell them many times. One person he did tell many times and did think it was important was his wife, Tula Hazelrig. And in fact, it's her, who is also in Columbia University, who really made the contribution that is how we use GFP all the time now. So here's Tula Hazelrig with her husband, Martin. This is obviously a moment when they get the Nobel Prize what she did then was take the gene for the green glowing jellyfish protein, attach that to a gene of another protein, and then you make a green glowing version of the protein that you are interested in following. So that is, of course, how most of us use GFP nowadays, to be able to visualize the position of individual types of proteins that we're interested in tracking. So it gives us a biological laser pointer, as Discover magazine called it at the time. So, Martin Chalfie wins the Nobel Prize, along with Roger Chen, who found other colors of that kind of protein from coral and also made all kinds of fluorescent molecules change according to things like calcium concentrations. At the time they won the Nobel Prize, Doug Prasher who, remember, he isolated the gene of the GFP protein and sent it to Martin to do the work. And he lost his job at Woods Hole. He then took a job working for NASA, but NASA changed their priorities and that project fell through. And in his words, he suffered bouts of depression and was essentially unemployed for a while. He didn't want to move to another place in the USA because he wanted to stay in the town where his daughter was finishing her high school education. So he was working on $10 an hour in a Toyota car dealership when these other people won the Nobel Prize. Doug Prasher says that he does not begrudge them winning a Nobel Prize, that they did the important work from showing the uses of the gene that he identified. Remember, this is a story about how we're going to get a microscope that I'm going to use in my lab that is beating a fundamental law of physics. So we'll get to that part now. This is Eric Betzig and Harold Hess. They worked in Bell Labs, which is famous for a very go-getting, intensive atmosphere, known for the discovery of the laser. Eric Betzig is shown in the blue shirt here, and this is his friend Harold Hess. Eric Betzig was working in Bell Labs, also developing microscopes, but he'd had enough of science. It's too hard. It was such a hard slog. It's too intense. He was working, incredibly long hours, so he'd quit. Age 42 or so, he was walking his daughter in a stroller, unemployed, when he had an idea for a new kind of microscope. He thought that could beat the fundamental law of physics that stops microscopes getting better and better. He published the idea of that microscope, but left it in that. He then met up with his mate Harold, and they were met up in national parks around the USA, and they were chatting about science. They realized, it was a bit of a shame they're not involved in science anymore. It is quite exciting. We do kind of want to get back in it. What is our legacy anyway? He then read Martin Chalfie's paper in Science about GFP, and in Eric Betzig's works, his jaw was hanging down for a week, and he thought, oh shit, I've just got to get back into microscopy. He said to Harold Hess, why don't we build the microscope that I had the idea for and published a while ago? They each spent $25,000 of their own money. They said, we could have renovated a bathroom, but what's the point of that? And they decided they're going to build the microscope that Eric had the idea of. They decided to build it in their living room. It was actually in Harold Hess's living room. In Eric Betzig's works, that's because he wasn't married. This is the microscope that they built on a cardboard box, all the stuff they bought with their own money, plus they sort of stole a few things from Bell Labs, as I understand it. And it started to work fairly quickly. Actually, they worked day and night. It was the sort of Bell Labs mentality back again. Eric told me that, quite often, though, he'd have a little nap and get Harold to carry on with it. This is how the microscope essentially works. So with a normal microscope, this is the surface of a cell, and you're looking at the fluorescence of one particular type of protein, and the kind of zoomed in part of that, you'd get some blurred image of all the proteins together. Eric's idea was that if you use a very low level of laser light, you would get a few of the molecules to shine on at any one time. And although you would get a big flash of light for where a molecule was, it came from one molecule because you only turned on a few of them at any one time. So you get a big flash of light, the fundamental law of physics is not broken, but you put a dot in the middle of that flash of light because one molecule is in the center there. And you do that iteratively, and then you can build up a single molecule picture of where every individual protein is. So it's not that they changed, a fundamental law of physics, but they found a trick of how you could get around that to make a microscope work better than anyone had imagined could happen before. It's the same as a helicopter does not change gravity, but it uses a trick to beat the fundamental law of gravity. Right, they realized that to get that demonstrated to work for the scientific community, they need to sort of get some reputation and do it with proper cells. Eric Betzig was going to give an talk at the NIH, and he knew that Jennifer Lippincott-Schwartz was there and that she had a track record of using microscopes to study cells. So he told Jennifer Lippincott-Schwartz, you've got to come to my talk. Jennifer Lippincott-Schwartz was busy, so, oh God, what is this guy telling me to come to his talk? Okay, fine, I'll go along. And within 5 minutes, she said in her words, he was a whole different other level of scientist, and she was completely blown away. It's really down to her brilliance as well, because she is now listening to a guy who hasn't published anything for 10 years, is unemployed, and instead he's built a microscope in his mate's living room that beats a fundamental law of physics. And she says, oh, okay, that sounds quite interesting. Eric Betzig, then between them, they agree to rebuild the microscope in Jennifer Lippincott-Schwartz's lab, which was in the NIH. And Eric and Harold both said that was, much less comfortable and less fun than it was doing it in their living room. This is the microscope that they built. Within 6 months from there, they got all the data they needed to win a Nobel Prize for a new kind of microscopy. I have skipped over some other aspects of this in the sense that they weren't actually doing that. The way I've just told you the story, it sort of sounds like they did it on their own. But in fact, other people were doing that similar kinds of things to build new kinds of microscopes, which I've written about before, but we don't have time to go into. But it wasn't that they did it solely without other people also doing similar things in parallel, as very often happens in science. So we are using these microscopes in my lab to open up, hopefully, new worlds about how immune cells recognize signs of disease in other cells. Some of you will know then that when I first started this, coming into the field from physics, we started to visualize how immune cells interact. And one of the things that we observed was that the proteins that you well know are important for the way immune cells detect disease in other cells, they accumulate up into patterns at the contact that immune cells make with other cells. And that came to be called the immune or the immunological synapse, because it was similar to what was known to happen between neurons, that there's a structured patterning of proteins between two cells. Now, essentially, we have a pipeline to visualize the single molecule resolution, what's happening at the contacts between two cells. So essentially, we get coordinates of the individual molecules. We can color code them according to how many other molecules are within a local radius. That then allows us to create sort of heat maps of how clustered things are. We can then define thresholds, and then we can really quantify the numbers of proteins in nanoscale clusters, how many there are, the density of them, which other proteins are co-localized, and that gives us a kind of very different view of how molecular recognition happens. I think that this is now opening up across every stage of how an immune cell interacts with another cell, so that the initial contacts are made with small protrusions, and then that have only recently been visualized. Effector functions have nanoscale structures to them, and then the NK cells detach. All of these processes are happening in complex ways on a nanoscale. For example, unpublished work by one of my postdocs, Jonathan Warboys, shows that an inhibitory receptor, TIGIT, dramatically relocates right to the T cell receptor to cause inhibition. One of the areas that we're particularly excited about now is imaging the secretions that each individual cell makes. So these cells are put on a slide, then you take the cells off, and where the cells were, there's essentially a dark spot, a black hole, a shadow, and then we can visualize at a single cell resolution everything that the cell has secreted in response to whatever is coated on the slide to stimulate the cell. This then has led us to see that perforin, that you well know is used to kill cancer cells or infected cells, does not come out as individual proteins, but comes out in protein clumps that's surrounded by thrombosbondin 1. Imaging macrophage secretions, for example, is our most recent work. And so essentially we have macrophages landing on slides. We take the macrophages off. We look at what every single cell has secreted. We use super resolution microscopy to visualize the exosomes, vesicles, and proteins that are secreted. We then analyze them according to different types of macrophages that we've derived from human blood and directly from lung tissue. And that gives us a completely different view of the types of macrophages that exist. So this is, you're not supposed to look at the details here, but essentially we're using these heat maps to characterize macrophages according to what they secrete. And there are 5 different classifications of macrophage that don't necessarily directly correlate with M1, M2, M0, or whatever cytokines they're known to secrete, according to the exosomes and other compartments that they secrete. And I think this is just the beginning of this. We're going to do this for other cells. Essentially, the single cell secretions can define functional outcomes of immune cells that are different to what we've known before. We've previously published that with the nanoscale clustering of proteins at the surface, activating receptors on NK cells called NKG2 co-localize with IL-15 receptor when they're being triggered by a tumor protein, LBIII. And the important point, though, is that these rearrangements in the nanoscale proteins at the cell surface actually lead to direct changes in the functional outcome, because that rearrangement of IL-15 into the NKG2D cluster synergizes with the signaling and gives a dramatic change in the exosomal secretion coming out of NK cells, which you wouldn't have known had we not done the nanoscale changes in the surface that lead to those functional differences. And this is unpublished work by Cordor Hazimi, who's sitting right at the back there. He's not allowed to escape. He'll take the questions. I'll skip this. This is essentially other imaging that you can only get these results from imaging. People thought that losing immune cell surface receptors was important for dampening immune responses. But in fact, it's important if you make a mutation so the receptors cannot be cleaved, immune cells just get stuck on the first target they meet. They have to cleave off receptors at the contact with another cell to be able to detach from it. And that is a druggable process. This is an antibody. This is an NK cell killing a cancer cell. And it's going to target this one, but then it's going to move on to this one, and then this one, and then this one, as you'll see here. So it kills one, then kills another one, and then it moves across to kill another one, and kills another one. And that happens because we've made an antibody that's very efficient at removing the cell surface receptors at each individual contact. I genuinely think that each of these new technology, I've just talked about microscopy and opening up new worlds in cells, but you will know that there are all kinds of new technologies being developed in biology. For example, single cell sequencing that many of you are working on, and Tracy also presented. Advances in understanding the microbiome is another good example. Many of you also working in neuroscience using all kinds of novel technologies. The point is, for me, is that it's exploding right now in every single aspect of human biology because of the advances in technology. And I think we're in exactly the same position that physics was in 1887. And that was when Heinrich Hertz, a German physicist, found the way to produce waves that we cannot see. Heinrich Hertz died at age 36, and he could not have known that the waves that we cannot see, radio waves, for example, TV waves, whatever, they led to the internet, the TV, the radio. He could never have foreseen that. And I think we're right in that position right now in biology, that we are advancing technologies that give us new views of how the human body works. They are unprecedented. It doesn't come from necessarily hypothesis-driven research. It comes from pushing technologies, and you open up new worlds of what the human body really is. And we have no idea how that's going to affect us in the future. But what I do think is going to happen is it's going to change the way in which we have to deal with very complicated issues. For example, if you do single cell sequencing and you are looking at the nature of all the cells in the human body, that is going to lead to any number of new ways of categorizing health and disease, and you will have to decide how much of that information you want to know for yourself. In the same way right now, we use the body mass index, and people are nervous about labels such as obesity or being thin. We are going to have an infinitely more complicated world to have to deal with in medicine. And it's really important that as biologists, we tackle those complicated issues as well as just doing the science. Physics, of course, in 1887 didn't just lead to TV, the radio, and it also led to nuclear bombs. And we have to be cognizant of where all this biology is taking us. These are all the people that did the work, but I also want to end with one other thing, very brief, don't worry, Mark. And that is, people ask me, why did you want to move from Manchester to Imperial College? And this is something that I've thought about very often. It's something that inspired me right since when Eric Betzig originally won the Nobel Prize. And essentially, when Eric Betzig won the Nobel Prize and gave his Nobel Prize speech, he ended by thanking all the people that got him to where he was, like every Nobel Prize winner. But then he said something else. And I just want to end by reading to you what he said at the end of his Nobel Prize winning speech. The last thing I would like to say is about taking risks. People are always exhorted to take risks, and that's fine. But you are hearing that from guys whose risks paid off. It's not a risk unless you fail most of the time. And so what I'd really like to do is I'd like to dedicate my talk to all of the unknown people out there in any walk of life who has gambled their fortunes, their careers, and their reputations to try to take a risk, but in the end failed. I'd just like to say that they should remember that it's the struggle itself that is its own reward, and the satisfaction that you knew that you gave everything you had to make the world a better place. Okay, thanks for listening.